I'm actually going to put that in. All right, welcome everyone to the Hatfield Marine, our weekly Hatfield Marine Science Center virtual research seminar. We'll wait a little bit longer for folks to log in and sign in. Just wait a couple more minutes here. Yeah, we got people filing in now. Great. Okay, we'll give it one more minute. I'll start. All right, let's go ahead and get started here. All right, welcome everyone. Thank you all for joining us today for the HMSC research seminar. My name is Yi-Chung Chung. I'm the academic program manager here at Oregon State University's Hatfield Marine Science Center here in Newport, Oregon on the Central Oregon coast. And I, as well as my colleague here, will be hosting today's talk. Just some basic logistics. We have your mics, cameras, and screen share off, and we ask that you keep them off for this event. Please ask any questions you might have using the chat box found at the bottom or the top of your Zoom screen at any time, and we will answer any questions at the end of this talk. We'll be recording this event, and it will be posted in a few days on the HMSC website's past seminar page, which I'll post in our chat right here. And will also be found on our YouTube page as well. Just some quick announcements to everyone. Just a reminder that later this evening, following today's seminar at 6 o'clock Pacific Standard Time, we have our next Science on Tap with Erica Fleischman, the director of the Oregon Climate Change Research Institute at Oregon State University, talking with us about last summer's wildfires and how science and society can respond to the changing dynamics of wildfire. Also, I wanted to announce next week's seminar speaker on May 20th will be Rebecca Chuck, David Tom, and Sam Derenbacher from the U.S. Fish and Wildlife Service Newport Field Office, who will be talking about public perceptions of coastal resources and marine protected areas, the Nestucca Bay National Wildlife Reserve, research and restoration intertwine to save an imperiled butterfly. So if you need more information for the links to our upcoming events, you can check our HMSC homepage and scroll to the bottom for more details. And that can be found here at this link that I just put in the chat. And now on to our honored guest. Today's speaker was invited by David Thompson, our resident restoration ecologist with the U.S. Fish and Wildlife Service, the Newport Field Office, as he aptly names himself, the resident guy who watches our grass grow. So with that, I'd like to hand it off to him to introduce today's speaker.
Thank you. It gives me great pleasure to introduce a friend that I have had the pleasure of working with for over 10 years. For over 25 years, Brian has focused on conservation through the geospatial sciences. He has worked with a variety of governments and their agencies, including some of the work that will be highlighted today. And he has worked for several universities, including teaching at UC Santa Cruz and running their GIS lab. And he's also worked both for-profit and non-for-profit organizations. His early projects included mapping paleo shorelines and redwood forests, designing ecological reserves, and predictive vegetation modeling. In 2008, he started working in San Francisco Bay and with me on mapping the intertidal zone from mean lower low water in their mudflats through the tidal marshes and into the uplands, searching for conservation opportunities for a future with climate change. Thanks for being here, Brian.
Thank you, David. I think I just heard a seagull. That was a nice introduction. So now I got to share my screen. So just give me a second. Click these boxes. Okay, that's, I think, a good introduction. And we have a slide. David gave a description of some other work we've done together in the Bay, and I have one slide that'll look at that. But the title of today's talk is Mapping Marshes and Mudflats from Space, Methods, Challenges, and Results from the South Bay Salt Pond Restoration Project. And, you know, I want to thank all the sponsors and the people who have funded the work over the first phase and now in our second phase of the project. We couldn't be here without them. And as David has already mentioned, you know, he is was the co-PI and really the lead biologist on all the project. One thing I'll note, I was joking before that the pictures, it's all down from here. This is the coolest picture I think we have, courtesy of Chris Benton, retired architecture professor at UC Berkeley, who has taken kite photography of the restored and non-restored salt ponds in the San Francisco Bay. This is one taken from A21, which we're going to look at. You can see the tidal marsh forming in the channels within the pond, the restored pond. And you can also see the ghost town of Drawbridge, these buildings sort of, which are right next to the Amtrak train tracks. I'm just noting that. If I keep talking too much for each slide, I'm never going to get through all of them. But a little background about the restoration project. You know, the San Francisco Bay has lost up to 85% of its historic tidal wetlands, and that's caused a severe reduction in marsh-dependent fish and wildlife. And so the South Bay Salt Pond Restoration Project, it started in 2003, is a 50-year project focusing to restore and enhance about 15,000 acres of former industrial salt ponds to tidal wetlands and other habitats. We're gonna focus on the tidal wetlands habitat, because that's the focus of the mapping. There are other components of the restoration, like enhancement to ponds that give birds nesting areas and other things. So I stole this straight out of the South Bay Salt Pond Restoration Project's website. It sort of thought to myself, well, people are gonna know how well it's been going. And I think this is even a little old. You know, the original goal was to restore about 50% of the land to tidal marsh habitat. I think it's 50 to 90%, depending on its impact on birds and other wildlife. But at the time they made this graphic, there was about 3,000 acres restored, so they're doing well. There are other things in the restoration, as I mentioned, including public access, which is amazing, flood risk, and then also enhancing some acres for wildlife that they've made good progress on. But again, we're really going to focus here on goal 1A, which is the tidal marsh habitat restoration. And so, you know, I had to put in some cute animals, but you know, the tidal marsh is an important place for endangered wildlife, such as the salt marsh harvest mouse shown here, and the Ridgeways Rail, formerly the Clapper Rail, and among many other important things that it provides the estuary and the flora and fauna in that estuary. And as we found out, as I did in the progress of this work, things like mudflats are a much more important food resource for foraging birds than we previously thought. Okay, so where does satellites mapping from space play a role? Well, the restoration needed a good method to track the evolution of the marsh habitats over the course of the project. And so David and I got that project to track that evolution, focusing on the vegetation and mudflats. So the goals of our project, entitled the Habitat Evolution Mapping Project, is on mapping the salt, brackish, and freshwater marshes within the study area, which we'll look at in a second, and with an important focus on mapping the floral colonization of the ponds that are restored, and also to track the mudflats, the extent and distribution of those mudflats, and as I mentioned, the presence and distribution of biofilm as best we can. And part of that is because not only do the mudflats play an important role in as a feeding resource for foraging birds, there was concern that as they open up the levees to tidal action, which is the main form of restoration that's done for the project, which is some passive restoration. It's opening up these levees and allowing the tide to restore the pond. There's a lot more to it. That's a very simplified version. There was some concern that is it going to erode the fully exposed tidal mudflats to the bay? And here I'm going to jump to the chase. I haven't seen evidence that it has. So that's good. We're mapping some other things. We map abiotic and upland habitats. And we have the data to map channels and just haven't done it, but there are other things that we produce from the project itself. And so finally, a map. So here, this is actually the satellite image we used for the project for 2019. And it says 2021, it's incorrect. I thought I had replaced it, I apologize. But the study area is in the South Bay of San Francisco. And it's everywhere south of the San Mateo Bridge, which is this line here on the top left of the image. The restoration has three main areas. One is Eden Landing up in the northeast corner. One is the Ravenswood, sort of on the western central side, on the other side of the smaller Dumbarton Bridge, and then this Alviso unit down in the south. We're not mapping just the marshes that are within the restoration unit. We're mapping every marsh, tidally exposed marsh, and even muted marshes, as well as the mudflats south of that bridge, because as we know that the estuary is a interconnected system, and so there's concerns not only about things like the mudflats, but also how is the rest of the system doing as we restore these ponds. You can see here in the center, there's still active salt ponds, these sort of green ones. And I'm showing this in false color, right? So these green ones are, where I was looking at it, would actually be red, which is the brine shrimp that are part of that crystallization process. So these, all the other ones, especially the ones within the yellow boundaries of the restoration, were purchased by the federal government for the restoration project. And I think it's still the largest wetlands restoration on the West Coast. I mean, one thing I like to joke is we're taking these very colorful ponds from space and turning them into something that's less colorful by the naked eye. But that's the nature of the beast. So what have we already done? So really I'm going to provide an overview of the two phases of the project and then an update to our preliminary results for our first update in 10 years. The first part of the project was in 2009 to 11. We successfully mapped about 15,000 acres of tidal salt and brackish marsh. We did not capture the image at a good tide at mean lower low water, and so we can't really use the mudflats we mapped from that era as a baseline. But in 2016, we were given research money to do a pilot study to develop methods to do that, which we did successfully. And that's what you're seeing here in brown are the 18,000 acres of tidal mudflats we've mapped using satellite imagery, and we've now reproduced in 2019. And as David more eloquently than me, and David made this map, by the way, I'm not going to take credit, The idea is really in that time period, we've also mapped the upland transition zone with sea level rise, which is sort of this blue band you see on the edge. And although I'm not going to talk about that work, David is very heavily involved in that. And we've really mapped the entire intertidal zone from the mudflats through the marshes all the way up to the uplands. So we get a really a good view of the baylands in San Francisco Bay and their current status and how we can help conserve and manage them into the future. In 2019, we began an update to that original habitat mapping project using high resolution satellite imagery. It was originally planned to be 2 years in 19 and 20, but a pandemic happened among other things. So we're doing actually the second year this year. We've already obtained an image and we're in process to create those 2 years. In 2019, I'm going to present you now the preliminary results for that. This is actually the first time I've presented those results. And that includes acreages of tidal marsh between the 2 periods and the acreages of mudflat between 2019 and 2016. And you can see the final data sets here. At the scale of the study area, it's very hard to see the habitats. They look a little green with a little speckles, speckles in different colors. and the sort of pinkish color over on the west side is mudflat with biofilm, but I'm going to zoom in so we can see this better. This is why I don't tend to show maps of the results often at the scale. But here you can see that study area, again, in the South Bay of San Francisco. And for those who don't know, this is really San Jose down here on the south. NASA Ames is pictured here. You can see the airport for NASA Ames, Silicon Valley, Google, Facebook. It's all within this picture, and I don't know exactly where because I forget. So I'm going to talk about the methods, and then we'll go into lots of cool pictures. Like I said, that first picture doesn't really get any better. So how did we do this? The whole project produced data sets using high resolution multispectral satellite imagery. And although not listed as a step, also including extensive ground truthing of habitat classifications, more so generally than we see with a lot of remote sensing. And why remote sensing? There's a lot of good reasons. One is we can capture the study area at one instance in time. We get multispectral data, which we'll talk about briefly, that allows us to discriminate between the different vegetation types. It's reproducible. It's cheaper than other imagery types. Everybody says, why don't you use drones? It's almost literally impossible to do this without in the wide area without a drone that flies higher, and getting airspace is difficult in that area. So there's a lot of good reasons why satellite imagery is a good pick here. And also tide. We capture the whole area in basically an instant in time. Although there's tidal variability, you can't really get that with aerial photography. So that's why we use satellite imagery. How do we actually produce output that's usable? The first step is to develop the vegetation classifications or habitat types that we're going to use, mapping vegetation, which are proxies for these salt, fresh, and brackish marshes. We spent most of that time in the first project doing ground truthing. That's David and his team to develop what we became the dominant vegetation classes that we used in the study. So we didn't do a lot of remapping for developing those classes. We sort of reused the classes from last time. So once we have those, we acquire the satellite image. It's a lot easier said than done because we want to capture the satellite images at the lowest tide, at mean low or low water. And because the satellite only passes over approximately at noon, it's never exactly at noon, and it does that to minimize things like sun glint, we have to match mean low or low water at noon, and then we want to, not written here, we want to do that at the time of sort of overall best phenology, which optimally is like June or July, and we've expanded now to May to August because of the difficulty of getting an image in that window. Oh, sorry. In our newest mapping, we got an image from the WorldView 2 satellite, which is an 8-band sensor, about 0.5 meter resolution. In the first project, we used the Icono satellite, which was a 4-band sensor at 1 meter resolution. And I'll talk a little more about the satellite. Once we get that image, we run a supervised classification using training sites. And that's what we call our habitat model. So that allows us to use the spectral values from the image and connect them with different things like pickleweed and cordgrass, alkali bulrush. And we use training sites, as I'll talk about, to supervise the classification of the entire image. We do that in an iterative process. And we also have a separate model for mudflats, which is a mix of methods designed to optimize the extraction of the extent of the mudflat under tidally variable conditions. And ultimately, we validate the model using a stratified random sample. So what are these habitat classifications? Here's some pictures of the, mostly taken with David 10 years ago, which I did not update those photos. But we're mapping basically 19 habitat types. We have 11 habitat classes in salt, brackish, and fresh marsh. We have basically three upland classes. Here it's listed alkali grasses, ruderal. We kind of have a broad ruderal or peripheral halophyte collection, but also pepperweed or Lepidium latifolium, which is here in the middle of this table, is sort of an upland class that invades the brackish marsh mostly and somewhat the salt marsh. And we have a number of non-vegetated classes, crucially mudflat with biofilm, which gives some idea of the distribution of this biofilm on top of the mudflat, microphytobenthos that exists that serves as an important resource for foraging birds. Okay, and because we map at this level here, we can sort of scale up if we need to, and this helps interpret the data. You know, we just want to know what's the status of salt marsh, or what's the status of low salt marsh or mid marsh. We can do that based on our classification scheme. So we have a pretty well developed habitat class. So for those who are interested, I oddly don't tend to talk much about the satellite, but the satellite is from now a company called Maxar. A great name, I guess. They really have the most advanced commercial satellite systems out there, and they really provide a lot of features for conservation. There are 8 spectral bands on the WorldView 2 sensor, and crucially, it includes one of these things is the coastal blue band, which is being included more and more on sensors, and it's on Landsat 8. It's a small slice of the blue spectra that allows us to peer through shallow water, which really helps us extract out those mudflats. And then also the yellow band is good for that, which is a unique band that's unique to the sensor. And for the vegetation discrimination, we have not only a red band, but a red edge band and 2 near infrared bands. So the spectral resolution really meets our needs. Basically, we pan sharpen the imagery, and so we get about a 0.5 meter spatial resolution, which is perfect for decision making. And it has a really good repeat time. That is, it passes over 1 to 4 days. But as I'll talk about, that you don't really get that access to the imagery because it's a commercial satellite company. I show here the Icono satellite, which was the first satellite we used. It's a lot coarser, a little older, but did the job at the time. So I have way too many slides, but I thought throw in some cool pictures of the results of 2019. This was taken on June 8th. I was at the Gorge that day. These are all false color image. So they are showing near infrared red and green. The bright red really shows us healthy vegetation. And so these are all ponds that have been restored, and we're going to look at the results from. And you can see, like on the top left here, A17, David had a site, didn't you, Dave? I think he built mounds on the east side of this levee for upland restoration, just related work. But the restoration is passive. They open up this levee, as you can see on the top left here, to the tide, and they sort of let the tide do the rest of the work. You can see those notches here in the middle, this A6, which we also call the duck's head because it looks like a duck's head. On the west side, there's a notch, and the east side, there's a notch. And you can see this in the Obamberg track here on the right, which is in Eden Landing. That was actually restored just as the project began. I think it wasn't technically part of the restoration itself, but a connected restoration. This one on the bottom left is not being restored to tidal marsh. It's used to show there's bird islands on them. You can sort of see these cone-looking shapes. But what we're seeing there is bottom reflectance of the old salt ponds, and they use it for at the end of a hiking trail. It just looks neat. So I thought it completed the slide. Okay, so once we get a great image, we run our habitat model, and we're basing on this tried and true method of supervised classification. And this image here on the top right is our training sites. The real first step to do that is we create little polygons around locations that are have usually more than 50% cover for each habitat type, let's say pickleweed or cordgrass. And it gains a spectral signature for that across the different bands of spectrum in the satellite image. And then we take those training sites, an initial set of training sites, and we run it through a statistical function. In this case, we used maximum likelihood that says for every other pixel in the image, or every pixel in the image, how likely is it to be in one of these training site classifications, one of the matching the spectral signature the most. So it runs through that algorithm and produces a result. And you can see on the top right, you see in the colorful ones on the bottom are mostly kind of classic vegetation signatures, except the blue water is a kind of a classic signature where a lot of near infrared and red light is absorbed, while in plant vegetation, it's rejecting that light, and we see higher values on the y-axis. And then above that, we have some of the abiotic classes, or not abiotic. In the case of alkali grasses, we have a classic grass signature, which is sort of almost like a linear shape. So you can see the differences between the different habitat types. So once we run that algorithm through, we go through a process of iterative improvement. We review the output. We send crews out in the field to say, hey, are we right? No, you're wrong. We look at it with other imagery and other data sets. We modify the training sites and rerun the model. And we do that iteratively until we feel pretty good about the results. And then we do an accuracy assessment. And then we assess the results using a stratified random sample stratified by the habitat classes that we use. Actually stratified by the salt, brackish, or fresh marsh, the relative proportion of those. So those accuracy assessments are based on pretty extensive ground truthing. So our ground truthing is based on the California Native Plant Society's rapid assessment or releve method, which is based on species dominance, codominance, or subdominance. It breaks up species and vegetation into plant alliances or plant associations. So you might, maybe not a good example, but like in a redwood forest might be an alliance and then an association with bay. So you have redwood bay as a dominant association. What's recorded on that form is species, the abiotic habitats, percent cover, pattern. We assign a habitat class and we do extensive photo documentation. The validation differs from our calibration fieldwork because the validation is at specific points randomly generated. We do a standard 20-meter radius and we assess ocularly those fields that I just listed. With calibration, we're more flexible. We're really just trying to check what's going on. And so we don't just do radiuses. We might do a linear feature. We might do polygons. It's fun fieldwork. In 2019, we went to approximately 295 locations. We had 180 validation points and about 115 calibration. The calibration was done mostly in 2020 and 21. And all of the validation was done the same year as the images acquired. You can see we had a pretty good spatial distribution of the data. A lot of remote sensors would say that's way too much, but we feel pretty good about that work. Then we put it into this error matrix, which basically compares our values in the field, assigned habitat classes, to what the model output says, and it gives us an idea of accuracy. In this case, our overall accuracy at the alliance scale was 82%. We're very happy with that result. And so we got 82% accuracy in 2019, which was higher than our previous years. And this is preliminary. It's preliminary because we will get data in 2021. And if there are any big changes that we see or errors we notice from the previous year, we might update the 2019 data. So we call it preliminary until the end of the second year. I'm going to skip the kappa, but it gives us some idea of how our classifier is it more than random? And a kappa really above 0.6 is considered pretty significant that it's definitely not random and we're mapping pretty well. I mean, the 0.78 was even higher than I expected. So we map the vegetation that way, but we developed a little different method to map our mudflats. The key part of that is obtaining imagery close to mean lower low water. We combine a series of indices and classifications to sort of get at the mudflats under this variable tidal condition. The first step is we run an unsupervised classification or an isocluster analysis that sort of groups like-minded pixels spectrally. We run that, and from that, we extract mudflat classes qualitatively. That is not difficult and easily reproducible in R and Python and Esri and a lot of different products. But we combine that with two indices or three. One is the normalized difference water index, which is a ratio of green to near infrared, which I'll show you in a second. And which is a good land water interface band index. And what we've done is we've kind of expanded that based on the literature to map water, shallow water, mudflat, and land. We add to that a third calibration measure, and that's one of two things. In 2016, we did an ordinary least squares where we regressed between the spectra of the imagery and bathymetry to map the mean lower low water line. We also noticed when we ran this other index called the modified soil adjusted vegetation index, which is a lot like NDVI, or the normalized difference vegetation index, which gives us an idea of chlorophyll absorption and greenness or health of vegetation. In this case, this one is really good for areas of vegetation that have a lot of ground cover exposed. So perfect for us for things like mudflats. We use that sort of to approximate the mean lower low water line, because we found such a correlation between the two. And then, we combine these indices as described here. It has to be identified as mudflat in the unsupervised classification and those other features from the NDWI. And then we use the MSavvy's zero contour to sort of calibrate how we're doing. And if we think we're not doing well, we might run the unsupervised classification again, or we might change how we classify the NDWI into the different types of water, shallow water. But in the end, it was a method that seemed to work very well and not hard to reproduce. So this is an image of the NDWI. You can sort of see the shallow water in the light blue. And then the mudflat is the green. I made the brightest green the mudflat. It really sticks out from the NDWI. And then many people say, well, we could just use this, right? Well, we use the multiple methods because it gets at places where there are shallow water that are missing from one but not the other. And we really found the combination works the best. This is just an example of the MSavvy. And then we have a result. We have the mudflats for 2019, which are shown in orange. People go, why don't you show them in brown? Orange sticks out more. And then that went too quickly. We have them in 2019 and very similar in 2016. Oddly, the biggest difference we noticed are within the restored ponds. So where there's significant floral colonization, we see a reduction in mudflat within the ponds. Okay. And we don't do a statistical validation of the mudflats, but we do extensive qualitative calibration, which is not easy. And so we walk, you can see here, we walked the mudflat at the time, at the exact same time of flyover or similar tidal conditions. And we see a high correspondence between our field data shown here in yellow, in this false color image, and with our extent of our mudflats. Really good calibration there. So we finished our work for 2019 and found some interesting things that since 2009 and 11, the vegetation and distribution is persistent. We've seen a slight growth of brackish marsh, which I didn't expect, versus salt marsh. Within the restored ponds, we see significant floral colonization. Outside of the restored ponds, on fringe marshes, we see a growth of low mid-marsh in some locations. Also of concern are invasives like Lepidium latifolium or pepperweed. We see a significant reduction in the pepperweed, but a persistence in its extent and distribution, and potentially a correlative growth in spear scale or atriplex. So invasives are still there, but the one that we've been thinking about seems to have been diminished to some degree, but it persists in those locations. Just seems a lot less dense. Okay, and so here's a table of our results, which is really just the quantification of those qualitative things I've said. I'm gonna go, I can get back to this if people have questions. Also, our mudflats, one of the big concerns here is we don't really see a big reduction in the extent of distribution since the 2016 and 19. Of course, that's a small time period. But we actually have a line of the edge of mudflat from 2005 from USGS, and there's also a high degree of correspondence. There's some locations of accretion and erosion, but those could be interannual variability. One thing I did say, and I noticed, is where we're restoring the ponds and there's a lot of floral colonization, we see mudflats sort of getting eaten up by vegetation. The other thing is we've noticed that although biofilm is probably everywhere on the mudflat, it's especially present within the restored ponds. Okay, so that's a lot of talking, and to get to these pictures. So what I'm showing here is sort of the results from the previous hemp 1 and our preliminary results for hemp 2. And this is one island that David has worked on here down on the lower Bear, or small island here on the southwest corner. This is all what we call Bear Island. There's outer Bear on the top, inner Bear in the middle, and then lower Bear, I think, at the bottom. And in 2011, I think there had begun the restoration of the lower Bear. And what we see here in this tan color, there's alkali grasses, more upland, pepperweed, lepidium, showing kind of a ruderal or environment. And I don't think they had fully exposed it to tidal action. The same is true for this gray area of inner Bear in the middle, which was more, which although was having some floral colonization, wasn't really developing fully yet as a tidally exposed pond. And then 2019, we see these are definitely developed into mudflats. The kind of tanner color is biofilm. So there's extensive amounts of biofilm on these mudflats with these restored ponds, but we don't see a huge amount of floral colonization difference between 2011 and 2019, which is interesting. The other areas in green, which are mostly pickleweed marshes or with cordgrass, they've remained very, very stable with very little change. And on the bottom, where they've opened it up to tidal action, we see the development of mudflats and some pickleweed along the channel. So some development there. There's actually also estuarine upland restoration going on there. Another pond restored, A6 on the left, or my left. The browner one is the 2011. They had just breached the levees. So the mudflats were forming within the pond. Not surprisingly, there was very little or no floral colonization. Jump to 2019, we see some development in green of pickleweed. The brighter green is cordgrass, but and it's starting to develop into some mats. But there's still extensive mudflat within that pond. But we have noticed some different changes at the top of this duck's head, the marsh on the top, which is outside of the levied pond. We see a growth in the marsh, and it's kind of hard to see on the top here. These are basically the same areas. We see the marsh kind of sneaking up on the bottom, and by 2019, it had grown about 30 meters of mostly low marsh or cordgrass with some alkali bulrush. And so, there's definitely the growth of marshes, but not always just in the restored ponds. This is the restored pond from the beginning, the A21 with that very cool kite photo. This is 2011. It's breached down here on the southern end. There's a smaller breach and a larger breach. We have floral colonization along the channels in green, a lot of mudflat, right? Uh-oh, wait, this was my fancy one. And by 2019, orange is alkali bulrush. We have an extensive alkali bulrush marsh formed within A21, and with pickleweed and cordgrass finding its way. Now, what's interesting is that this marsh seems to have developed relatively quickly and is weirdly seasonal, which goes against all thinking, which I had to actually check with multiple sources, sort of check the work. The other thing to notice is drawbridge, that ghost town is in the middle, and this line here, remember this is preliminary, this kind of blue line on the right side is actually the Amtrak train tracks, which we have to mask out. But you can see where that whole ghost town was. This area is still, the red is sort of pepperweed invasives, but it seems less dense, could be phenology. It's there, but it seems less persistent. We move east along that creek, we go to another pond, we see reducing, the salinity seems to go down as we move east. On the right here, this kind of orange mosaic is alkali bulrush, so the east of the pond, with red of that invasive pepperweed invading the marsh. It was mostly mudflat after the breach in 2011, with some spots of floral colonization. I'm trying to be fancy. And then in 2019, certainly it's still mostly mudflat, but interestingly, this purple is a large bulrush or freshwater bulrush marsh that's developed. And how do we check our work? I'm like, that's got to be wrong. I talked to the field guys. Yeah, that looks wrong. And then we get word there's a huge water bulrush marsh out there in A19 that's developed really over the last 3 or 4 years. So it's unexpected calibration with colleagues. Certainly we see some colonization of pickleweed too, but not as extensive as the other A21 pond. Other places outside of the restored ponds, we see the flora colonizing. This is Ogilvy Island, which, if I'm correct, David Thompson helped name. And on the top, we have 2019. It's mostly mudflat with biofilm. And then by 2019, we see on the bottom, it's all a giant pickleweed and cordgrass marsh with alkali bulrush and orange sort of moving into its territory. More examples of growth of marsh. This is another marsh outside of the ponds where we see a growth of low marsh moving southward about 40 meters between 2011 and 19. Other marshes, there isn't a lot of change. This is a marsh in 2011 that had a lot of high marsh, which is important for obligate fauna. We sort of see that in this pinkish stuff along the channels on the western image. It seems less in 2019, but that could be time of day, phenological variability. It's still rather pervasive throughout the marsh. The marshes look relatively similar. So that's also something we capture. In this case, we also captured more alkali bulrush than we saw at all in 2011, which is that permanent change or seasonal variability? It's hard to know until we continue to map. Another restored pond up in the Eden Landing area, which is the northeast, extensive mudflats. Pickleweed is forming into much more dense maps in the southern part than it is in the northern part. But we see the tidal marsh really starting to form here and salt marsh. So that's a lot of pictures. What are the challenges we had? One big one was timing the satellite overpass with mean low water at noon, which continues to be a challenge. And one thing I've learned, the company was bought since we worked with them last time. And this time they pretty much locked us out from working with them at all, because we really need to tell, if they could tell us when the satellite's going to fly over, we could try to find the best day, but they refused to do that. And really, we find we're a very low priority against very large clients like the DoD, which is a shame. Sort of standard challenges that there's confusion between certain habitats spectrally, but we work those out as best as we can. In the end, we have to manually edit some things. And then there's always phenological variability that could be inter or intra-annual. And one thing we're going to do in 2021 and 2022 is clean up all the noise to control for differences for our change analysis. And the background here is actually channels in Bear. And the imagery really is amazing, and the data we provide is amazing to show the channels within the ponds, and we really want to produce that as a data set this time. That's it. Does anybody have any questions? That's a lot.
Thank you, Brian. Did we lose you? I think we lost your video.
Oh.
We have a question from Ted DeWitt from EPA. Really great work. What is your confidence in being able to accurately identify species? And are the image classification algorithms likely to be directly transferable to other locations, assuming they were using world view imagery and have the same veg species?
That second one, probably, I mean, you can try to use them, but it's very, very hard to control for a lot of the factors. We used atmospherically corrected imagery, so we're supposedly getting ground reflectance values. So technically, you should be able to use the spectra signature files that we created to map other locations. But practically, I found over a 20+ year career that you always need to modify them. And that has a lot to do with that sensors like Landsat have better radiometric control. So they can create more widely distributed spectral libraries versus WorldView 2, which is an amazing sensor. There's still more radiometric variability. So the answer is sort of yes, but qualified. And then the first part of the question was, what's my confidence? My confidence is very high at the alliance level, and overall our accuracy was 82%. That varies on habitat to habitat, but I think the lowest accuracy level was like 64%, and which we hope to improve, but was also pretty good. So I feel pretty confident over the study area in the data that we produced.
Great. Any other questions from the audience? Feel free to post them in the chat, or feel free to turn your video on and ask that question. All right. I don't see any additional questions. And Dave, let's see here. Did we lose?
No, I'm here.
Okay, great. I don't see any additional questions.
Okay.
Brian, did you have anything else that you would like to add as far as?
It's, just because it's California doesn't mean we have to think it's bad. So.
Do you see this being applied to any other estuaries on the West Coast?
For sure. I mean, I think that whether we can apply the same spectral library or not, the same process is very repeatable. And I don't use black box methods purposefully. So, and I don't actually create additional complexities because we want it to be more reproducible. So the process itself, I think, is very reproducible along estuaries anywhere on the West Coast. So, that's the idea, is develop sort of standard methods that make it easier to reproduce over time and in other locations.
Now, here's another question. Have you looked at associating ecological function to the changes in the habitat features identified in the imagery, such as abundance of wildlife?
That's the long-term plan, right? So we haven't done that yet, but we really want to. So, the data will be used to assist in the adaptive management for the restoration. And the long-term goal is to look at the wildlife, habitat relationships and partly using the data sets. It's just, they have so much to do. It just, they get what they have to get done first. And then often that kind of research comes second. Very excited to do that, though. We are working with partners to help compile some of that information too, so we can compare the two.
Yeah, I guess my follow-up question to that would be: so are there management partners that are looking to utilize this for management in some of these areas?
Well, the restoration project itself, and we'll use it to inform the restoration. And the data has been used by a range of different people. It was used for a carbon sequestration study, for the wetlands, for a research group out of Colorado. Different flood control districts have used it. The Corps of Engineers have used it to inform their potential changes in the Palo Alto Baylands, which are within the study area. So there's been a variety of different uses of it, and the restoration project themselves have used it to sort of inform what's going on in one location, and they will use this update to also inform them. And so they haven't gotten it yet. So this is, like I said, the first time I presented the results. I'm presenting it to them next week. So we're excited to produce that for them. And what happens then is we give the data and give them access to the data, to the stakeholders, scientists, and researchers, and then they can use it at will. And also they help provide another calibration measure, because a lot of them are so familiar with certain locations, to inform us where it might be mapping well, or where there might be places for improvement. So,
I also had a kind of a question, just a curiosity question about the ghost town that you mentioned related to the train tracks. Is it just an old? Is that just a location of an old settlement?
You know, David, if he's still here.
Yeah, I'm here. You want me to answer that, Brian?
What?
You want me to answer that, Brian?
Yeah, you would know better.
Yeah. So Drawbridge was a settlement. I'm trying to remember when it got started, but it didn't finally become a ghost town until the 1970s. And a number of things led to its ending as a settlement. The amount of water withdrawals for agriculture in the Santa Clara Valley led to groundwater withdrawal and drew subsidence that reached 13 feet in 50 years. It was the first place I ever saw that kind of subsidence in the world due to groundwater withdrawal, and that caused, of course, some people that live out in the tidal marshes to lose their somewhat dry. And then also the population density in the area led to really bad water quality until the Clean Water Act started to fix that, and they were losing their shellfish and their fishing, and so things just became intolerable for them. that area. Yeah, like I said, not that long ago.
Thank you. All right. Well, looks like there aren't any more questions from our audience. So I guess we're letting you off early there, Brian. So
Yeah. No worries.
thank
It's a
you.
pleasure.
Yeah, it was a real pleasure. And again, just wanted to remind everyone, this is recorded and it will be on our past seminar page as well as our YouTube page. And later on this evening at 6 o'clock, we'll have our Science on Tap, which I posted in our chat there. So thank you everyone for joining us. Brian, David, thank you for presenting to us. And we really appreciate it.
No problem. Thank you.
Thank you.